Hey everyone, welcome back to our YouTube channel Savvy Forensics. So, in this video, we'll be moving towards the identification of bodily fluids and we'll be looking at the second biological evidence, which is semen. In the previous videos, we have looked about the identification of blood, its preliminary identification, its confirmatory identification, its origin of species, test, and so on. From this video, we'll be now shifting our focus towards the semen, which is also a very important biological fluid that is encountered at the scene of crime, basically crimes involving sexual assaults and related ones. So let's discuss them. So the identification of bodily fluids, that is semen. These are the table of contents we are looking. Firstly, we'll understand what is semen, its biological properties, as well as the major component, that is spermatozoa, that is used for identification, then the presumptive assays and the confirmatory assays. Now here I want to tell you that in this video we'll be covering these three topics only, and in the next video we'll be covering the presumptive and the confirmatory assays. So let's discuss further. Now, what is semen? Semen is a faint yellow colored fluid with characteristic odor, which is called as seminal odor. Now, a typical ejaculation releases 2 to 5 ml of semen, which contains seminal fluid and sperm cells, which are also called spermatozoa. So, these type of questions are many times asked in your exam. That these are basically fact-based questions, like what is the amount of semen that is released per ejaculation, or what are the approximate count of the spermatozoa that is present in per ml of the semen. So you can remember this. The seminal fluid is a complex mixture of glandular secretions. Now, why are we saying that it is a mixture of glandular secretions? Because various glands are involved in its secretion, which we'll be looking further. A normal sperm count ranges from, this is written on, it is 10 raised to the power 7 to 10 raised to the power 8 spermatozoa per milliliter of semen. You also have to remember this is also very important. Now, a semen consists of, semen or the seminal fluid, it consists of three major components. The spermatozoa, which constitutes 10% of the total seminal fluid, the seminal plasma, which is of 90% content, and the epithelial cell, which is less than 1%. Now, this seminal plasma, which constitutes 9% of the total seminal fluid or the semen, it further consists of the secretions of various glands, which are the seminal vesicle, the prostatic gland, epididymis, and the bulbourethral gland. Seminal vesicle fluid, which is secreted from the seminal vesicle, it constitutes 60% of the total seminal plasma. Then the prostatic fluid, which is secreted from the prostate gland, constitutes 30% of the total seminal plasma. The epididymis secretion, which constitutes 5%, as well as the bulbourethral secretion, it is also constitutes 5% of the total seminal plasma. Now, these particular secretions or the fluids from the particular glands, they consist of several specific entities that are used for the identification of semen. Like the seminal fluid, it consists of flavins, which usually when we drop alternate light sources for searching of seminal stains, they give fluorescence. So we get to detect that particular location consists of seminal stains. Further, the prostatic fluid, it consists of enzymes acid phosphatase, which is very important for the identification of semen, as well as the prostate specific antigen. We'll be learning all this in detail. I'm just telling you for a brief count that you just get to get familiar with these terms. The epididymis and the bulbourethral secretions, they also constitute 5% of the total seminal plasma. So let's move further. Now, the biological properties of semen. Basically, we'll be looking at the seminal. So the first is the seminal plasma, which constitutes the 90% of the total seminal fluid. It consists of testes, epididymis gland. Volume in ejaculate will be 0.15 ml, just 10%. Approximately, it contains 80 million spermatozoa. Again, the second one is the seminal vesicle gland. Its constituent in the ejaculate will be 1.5 to 2 ml, which will be approximately 50 to 65%. It consists of the components such as fructose, phosphoryl choline, argothionine, ascorbic acid, flavins, as I told you earlier, that they give fluorescence when they are irradiated with UV light, the prostaglandins, and the bicarbonates, major components. Then the prostate gland, it constitutes 20 to 30% of the ejaculate, which is roughly 0.6 to 0.9 ml. So it consists of prostate, spermine, citric acid, cholesterol, alkaline phosphatase, phospholipid, fibrinolysin, fibrinogenase, acid phosphatase, and the prostate-specific antigen. Major components that are used for the detection of semen. So from here you can infer that the seminal vesicle gland, it constitutes major portion of the ejaculate, just 50 to 65%. Let's move further. So let's understand some other biological properties, like semen is extremely good source of DNA. Why? Why it is a good source of DNA? Because it consists of a nucleus, right? And nucleus contains